recorded. And it will be made available in the SciLifeLab YouTube channel, and you will also be able to get the presentation through the SciLifeLab Share instance. Yes. Yes. Okay. So the agenda for today is that I will start here with an introduction, and then we will move into the overview of the Swedish Reference Genome Portal, where Daniel will have an introduction and a live demo of the service. Rory will talk about the technical implementation, and then we will have Angela who will show some features that boost and facilitate researchers' work. And if you have any questions, I would suggest you to please enter them into the chat, where we will have a look. And we will also have then a Q&A sessions at the end. So, all right. First, some information about the data-driven life science program, because the Genome Portal is made as a part of the data-driven life science program. So the aims of DDLS is to develop and operate data services and resources with high impact for data-driven life science in Sweden, to lead cutting-edge technology development to strengthen Swedish competence in the area. And SciLifeLab has appointed SciLifeLab Data Centre to organize the data support and databases operational area. So the vision and mission is then to develop and coordinate national data services and resources for the corresponding research area, and to provide these services in coordination with the SciLifeLab Data Platform. And to the right, then you have the funders and the interested parties here, the involved parties in the program. So the data-driven life science is a very, very big program funded by the Knut and Alice Wallenberg Foundation. It includes several parts, but at the bottom there in the green frame, you can see the WABI support, which is bioinformatics support, and then the data support, which are the two fundings that are then going into this data science node that we're working for. There are 4 research areas in DDLS, and each of these has now a data science node. And the service we are presenting today is being developed by the Data Science Node in Evolution and Biodiversity, which is hosted by Uppsala University, but we also have staff at the Museum of Natural History in Stockholm. Then we have the other data science node, the precision medicine at KI, epidemiology and biology of infection at the Umeå University, and cell and molecular biology at Chalmers. And for each of these data science nodes, there are 3 full-time equivalents working with data service led by Data Centre, and 2 full-time equivalents working with project support, and they are hosted by NBIS. So this is very much a collaboration between NBIS and the Data Centre at SciLifeLab. Here we have all the staff employed working in the data science node. So you have me there as a scientific lead at the top. We have Angela working as a coordinator at Data Centre. We have Quentin working at the Museum of Natural History as a system developer. Rory working as a data engineer at Uppsala University. Daniel working as a data steward at Uppsala University. And then we have Hanna and Johan, who are the vice head and head of Data Centre. Bengt Persson is formerly the lead of the data science node. And then we have Cormac and Miguel working as the bioinformaticians in the node. So Cormac is at the Museum of Natural History, and Miguel is at Uppsala University. So that's actually an error there. He should be at Museum of Natural History. The people that have been involved in developing this Genome Portal team is the people in the upper row there. So it is me, Angela, Quentin, Rory, and Daniel. Just briefly, there are a lot of reference genome projects going on now in the world, and all of these are in some way connected to the Earth Biogenome Project, which is called a moonshot for biology, where they intend to sequence and assemble genomes of all eukaryote genomes in the world. Around 0.2% of the eukaryotic diversity is estimated to have reference genomes at the moment. And you can also see that a lot of these species are in decline, for example, in the vertebrates there. So a lot of reference genomes are now being assembled, but they also need a place to live and to be presented to the research community, and that's where the Genome Portal comes in. So genome portals collect and help visualize data. And they typically consist of a set of web pages with information and links and a genome browser. And we will then see much more about this later today with Daniel's and Angela's presentation in particular. So we have developed a Swedish Reference Genome Portal, and we now have the official launch. The aims are to highlight and showcase genome research projects performed in Sweden. Species doesn't have to be in Sweden, but the projects need to be driven from Sweden in some way. We want to make it easier to access, visualize, and interpret genome data and lower the barrier entry to look at this type of data. We want to promote sharing of annotations of genomic features that otherwise would not be published, and we can also help in the publishing of this data. And we want to ensure that all data shown on the Genome Portal is aligned with the FAIR principle and available in public repositories. So now I will give the word to Daniel. We'll continue the presentation.
Yeah. Thank you, Henrik. Thank you for giving the high-level context of our project. And now we will have 3 talks from me, Rory, and Angela about more of the details. So if we start here, a slide about the scope. The scope is slightly aligned with the aims, of course, but this is more like what type of data are we willing to host or showcase in our portal. So we have, because we are DDLS Evolution and Biodiversity Data Science Node, we have them focused on non-human eukaryotic species, like Henrik was talking about with biodiversity. And the same for the next bullet here, that the researchers need to be affiliated to a Swedish institution of sorts for us to be able to host it. But other than that, we will welcome every single species that you may want to put here. And in terms of formats, we wanted to be a genome assembly, of course, but also at least a track with protein coding genes, because these feature annotations are so important to make biological interpretations of the data. So this is the minimum that we ask for. But other than that, it's up to every single researcher to put whatever they would like on the portal. So we are very flexible, and we'll try to tailor the pages to each of the research group's needs. That takes me to the last bullet, though, that we are not a repository, we are a genome portal. So we want the data to be publicly accessible. And there are domain-specific repositories for nucleotide data. ENA and NCBI will cover most of the formats, but there might be some tracks that we actually help you broker somehow. For instance, putting on the SciLifeLab Data Repository, where we can make your files publicly available so that you can share it in your manuscripts or with your peers, as well as we can pull it into the portal. So we are very happy to help with that. And should you also need help with ENA and NCBI, we can at least point you towards people who know more about that type of data sharing. But we offer this data repository service as part of the package of putting an organism in there, to say.
You can just waiting for good.
There you go. Now I have control. Right, thank you, Angela. So genome portals, they can be designed in many different ways, and there are many genome portals out there in the world. And our portal is obviously a multi-species portal, meaning that we don't just host one model organism. We accept all types of different species. But to be able to have some sort of skeleton for the work we're presenting, we have divided our portal in 4 different pages, as you see here. And I will demo soon live as well for you. We have a description tab where we have some information about the occurrence, biology, and taxonomy of the species, with information of how to cite the data, of course. We have a genome assembly tab where we have a lot of bioinformatics information, how was the assembly annotation performed. We have a downloads tab, which we hope will be very useful for everyone visiting our site, because we can aggregate links to all the different data tracks that people might have stored in different repositories across the internet. So instead of having to read data availability statements in scientific manuscripts and try to figure out where all the links are, we can collect them here in one place to make it easy for you to find them. And those tracks, they are of course also then showed in our genome browser. And some words about the genome browser. We are using an open source software called JBrowse 2. We have evaluated a few different options, and we have settled on specifically JBrowse. It's very nice for us because it's designed to be embedded in websites like ours. It also has a desktop client, so people can, of course, download every setting from our portal and run it locally if they want. And it's a very active community developing this software right now. So that means that we can easily get support. It's updated very frequently, almost every month, I believe there are new updates. And there's a lot of people working on making plugins for this software. So we think that this is the future for the genome portals in this context. And as Angela will show you later today, there are also some features that you can play around with. What you see here is a typical linear genome display that I think if you've used genome browsers before, this is probably what you have seen, different tracks stacked on top of each other, but JBrowse has more than this. So we hope that in the future we might be able to host more interesting, or not interesting, more advanced features as well. For those of you in the audience who are more like bioinformatically inclined, who want to know about file formats, we can say that JBrowse can support all the standard file formats. Here is a list that we picked of some common ones. There are even more than this. I think these will satisfy most needs. Most bioinformatic pipelines and tools will output in these formats or similar. We've actually been playing with similar to this as well, and we can get into display. It all again depends on your needs in your research groups. We have been playing recently with displaying population genomics data in a bed-like format, where basically the first 3 columns of the data is bed formatted, but the other columns in the data set are free up to the users to describe. So as long as we know what part of the data you want to display, we can tweak JBrowse to show that for you. So we think this is a very fun way to work together with you to customize the data you want to show. We have put a little caveat here right now that we have seen that BAM files do negatively affect performance, especially on our web server. So right now we are not putting any BAM files up on our genome portal, but that doesn't mean that you cannot use them as a user. You can actually load them yourself in the genome browser by clicking a button called Add Track. So on your client side, you can put any tracks you want, as long as the track you're putting is formatted towards the same version of the assembly. So if the coordinates match between your file and the assembly we have put on the genome portal, you're good to go. So you can play with any BAM files, and that could be very useful for certain analysis, right? We might look into the future to make some performance enhancing updates to this, but we will see what that will take us. It all depends on your needs, basically. So we'd love to hear from you what you would like to see as features in the portal. And with this, I think it's time to make a little live demo. So if we manage to close the presentation.
If you click, it should go.
Click escape. There you go. And I can switch here. And I hope everyone is with me right now. Into video. This is where you get, if you right now go to genomes.sciliflab.se, the site is live and fully operational. I hope some of you might even have played with that already. So you're welcome to just go here and poke around, but I will showcase some highlights. So this is where you get when you type in the URL. This is our homepage. And right now we have 6 different species that we have been very happy to collaborate with some research groups to get these pilot species in for our launch today. You can see they are from different parts of the eukaryotic domains. And you can click on these different cards and get the information. So just for the sake of it, I will click on this linseed plant today. And I will show you how it looks like. So here we start by having a little picture given to us by the researchers, and an interactive map that pulls data from GBIF. You can play around here and see where in the world have people reported this species. And we are pretty happy about this feature because we think that most of the genome portals don't necessarily present this data. So we're very keen to hear what you think about that. We would love to work more on that. Then we have some taxonomy information, some external links to harmonize with other websites. a species description. So if people have never heard of this particular species before, they can learn a little bit. There's academic references in place. And of course, very important for us, for the sake of open science and FAIR principles, is how to cite the data. The data, of course, belongs to the research groups, and here you will easily find the paper or papers that the data comes from. If you also want to cite our portal as a service, we of course would appreciate that, but of course citing the data from the research is always the highest priority. But we have a little citation for the portal as such as well. That's the description page. If I go down up here to the assembly page, you will get more like bioinformatics information. So those of you who are familiar with this type of statistics might directly recognize them, but we collect them here so that people can quickly assess the quality of the annotation and assembly for those who are in the know, basically. Let me repeat the data citation again over there. The downloads page I am particularly happy about, because here we then try to collect all the tracks in one single place to make it easy for you to access the different tracks. And for each of these tracks, you will have direct download links to the data, link to a website if there is any, for instance, an NCBI or a data repository page will take you there. And of course, the articles that you know what track belongs to what citation. It's even possible to extend this view to get a bit more information about accession numbers, DOIs, file names, and so on. We hope that this is going to be useful for aggregating data, making it easier to find data sets from a particular species like this. Then, of course, the crowning of all this is the genome browser, and I will open it here. Now, this one has many tracks. That's why I want to show it to you today. So we have to wait for it to buffer a little bit. There we go, as expected. So when you come in here, you will see JBrowse embedded in our browser. And depending on the zoom level that we have set and the data, you might get messages like this that you might need to click a button to initiate a load. So don't worry if you see warnings like that. We up here then, for those of you who are not that familiar with genome browsers, we have a coordinate system up here for this particular chromosome we are showing. So the nucleotide bases are here. You can even turn on the DNA sequence if you want. I have it turned off right now, but it's possible to get the DNA as well. Then the first track, the one that we always ask researchers to supply to us, is the protein coding genes with all the different features annotated. And if I zoom in a little bit, we could maybe click on a box just to show you how interactive it is. On the side here, we should hopefully get the details from this annotation pool from the GFF files. So depending on how the annotation looks like, you will get different information here that you can view also in the graphical interface. Let me go back to the tracks again. And in this case, the researchers have decided to put in some repeat elements and then a lot of population genomics data. See if we can scroll down. Here we go. So this data is pretty sparse. So right now, looking at this zoom level might not be that interesting, but I could either zoom out to full extent to see the data like this across the chromosome. Or if I change to show all chromosomes at once, we can see it stacked like this, making it, depending on your research question, and I'm not the author of this data, so I shouldn't make too much of the interpretation, you might then be able to compare different populations or different tracks and like visually inspect regions that might give you clues of new questions to ask. For instance, here you see that there are certain regions that are high in all of these population tracks on this particular metric. So maybe that tells you something that you can work with. So it could be a powerful tool for visualizing many tracks on top of each other like this. That is, in general, the genome browser. I would just like to end this little demo by talking about how you can get help understanding on how this page works. And up here in the right, top right, we have a lot of different navigation links to click. For instance, how do you go about to add your data to the portal? Then you can read about it here. Very shortly, we will help you as much as possible. Just let us know that you have data, and then we will contact you, talk to you about where is the data, how can we find it, what type of visualizations do you want, and we do all that work for you. We want this to be as easy as possible. So it's just about initiating contact, send us a mail, and we'll fix the rest, basically. If you need help with brokering of data, if you need help with formatting, we will make sure that we fix that for you. So make it as easy as possible, just contact us. But there is some text for you to read here if you're interested. Then we have a user guide, which is basically what I'm saying now aloud in the live demo. We have written that down. So there's a lot of text here to look at if you are wondering about certain parts of the website. We are particularly happy about our small guide to the genome browser. Because even if you are familiar with using genome browsers, for instance, you might have used IGV for many, many years, changing to a new software always takes a little bit of getting used to it. So we think that this could be helpful to take a look at getting to understand where the different elements are located in the UI. So there's some text for that as well. Then finally, we have a glossary for terms that we have used in the Genome Portal. This is about lowering the of entry for genomics, if you're, for instance, starting out in this field, or if you're wondering what do we and the staff actually mean when we use different terms. We have pulled in definitions from open source ontologies, so-called controlled vocabularies, where we bring in those definitions to make sure that whenever we say annotation track, this is what we mean. There's quite a long list here, so we hope that can be helpful for you. And we would also love to hear input if there are things here that we could add or maybe rephrase a little bit. We have some frequently asked questions if you run into issues. But of course, the easiest way is probably to contact us. So we have a contact form here or our email address. So don't be shy, just reach out and we will do the best to help you use the page, use the service, answer your questions and so on. So with that, I think that is roughly what the Genome Portal website does today. We will be very happy to hear from you how you like it and if you have any feedback and thoughts about it, feel free to talk later in the Q&A or send us a mail. So I will now close and pass over to my colleague Rory, who will give some technical details.
I can do it from this one. Yes, hello everyone. Just bear with us while we change who can control the computer. It should now be me. Okay, yeah, now I can. Good. Thank you, Angela. Yeah, so I will give a brief description of the technical implementation for about 5 minutes was the estimated timeline. So I will try to give a very high-level overview and then focus on one thing in particular because it's too hard to try and explain everything. But hopefully it will be interesting and approachable. So the first thing I want to say is that the source code for this project is all publicly available on GitHub. You're very welcome to take a look and you're very welcome to provide us with suggestions, ideas for new features. Although also if you see a bug, a typo, or maybe you see a correction about something that you think is scientifically inaccurate, etc., please feel free to get in touch with us. And if you're interested in contributing, you're also, whether that be code-based contributions or maybe other kinds of contributions, we're really very open. And as it's already been mentioned, there's plenty of ways to get in touch with us. So you can email us, you can use the contact form on the website, as Daniel just showed you. Or if you're comfortable with it, you're very welcome to get in touch with us directly through GitHub, whether that would be by creating an issue or whether that would be by creating a pull request. Everything is welcome. So just to give an overview of the different technologies that we've used to build this genome browser, or genome portal, sorry. We use a range of open source technologies, and that's strongly encouraged by the data center that we work for. And to build the website, we use Hugo, which is a static site generator. And when I say static in this case, what that means essentially is not that you can't have dynamic content on the website, so you can have animations or things like that. It more means that the website is already pre-built, and that means it can be sent to a user very fast, and that helps to provide a nice experience for a user. You don't have to wait as long to reload a page, for example. To make the nice kind of observation maps that we have, we use the GBIF API, but to display it, we use a library called Leaflet. And to kind of help style the web page, we're using Bootstrap. For the genome browser itself, as it's already been shown, we're using JBrowse as the genome browser, which is very easy to kind of implement into a website. But then in terms of preparing the data sets, because we are not the primary source for these data sets, instead it's the repositories that we can help you put your data onto. We use a combination of some bioinformatic tools like SAM tools. And then we also use a program called Make, which is a bit like an orchestrator. So it helps essentially take all of your input files and put them into the output files that you want. When it comes to deployment, we create we create applications inside Docker containers. And then we have these on our Kubernetes cluster, which is at Stockholm's KTH University. And then we also use and GitHub Actions quite a lot to help us kind of automatically test our code as well as automatically build those Docker images that we use in our applications. So rather than talk about like all of the different aspects, I wanted to try and focus on one thing and how we kind of approach this problem. And that is that we're doing software development, which involves working with large data sets. And so typically in software development, you rely on kind of rapid cycles of continuously integrating your code changes. So this is like a 3-step process called where you essentially implement some code changes, you rebuild the software, and then you test that the software performs as expected. If all goes to plan, then you just repeat this process over and over again. And what you want is that the build and the test steps to be kind of fast and smooth. You don't want to have to wait ages for those steps. And so this is the CI in CI/CD, if you're familiar with that term. So when it comes to working with these data sets, as we're not the primary source for these data sets, there's potentially a very big bottleneck when it comes to the website. And that is that we would first need to download these large data sets from a primary repository, such as the ENA. And then we also need to pre-process these data sets so that they can be shown onto the repository. So just as an example case for this, we have Northern Krill, which is shown on the website, and it's about 5 gigabytes of data that we need to download from the ENA every time, or 5 gigabytes of data that we need to download when in compressed form. And this also takes many hours of compute time to pre-process this data so that it's ready for the genome browser. So that can be unzipping and indexing the file, for example. But it may be the case that you might think, well, then we can just download it once and we're done with it. And that's true, but then once we make changes or updates or there's a new version involved, that's going to become very awkward, and we're going to have like some sort of manual track of making sure that we're always on top of that. Otherwise, we'll end up introducing bugs that will be very hard to detect because essentially a file has changed, but we haven't noticed. So in order to like get around this problem, Make, which is a program, which can be almost thought of as like a recipe book. So we create a set of recipes inside a Makefile that defines how we want our inputs to become our output. So if we keep going with this kind of recipe analogy, we can think of the configuration files as the code. The configuration files as the ingredients in these recipes, and the code as ways to kind of process the ingredients to make different meals. And what that means is that when we run Make the first time, we produce all of these different meals, which in this case is just different data files. But what's nice is that the second time we run Make, if we already have made this specific meal, we don't need to make it again. So that essentially means that if it's a very small update or only a small change, it's a very, very quick process the next time around. However, if the ingredients have changed, then Make is smart enough to know that, oh, okay, we need to update this specific recipe. And what this essentially means is that most of the time you have very, very quick build steps, which allows for very quick iteration of that continuous integration cycle. And so that's very nice. So if, for example, we had a change in one of the configuration files where 2 of the recipes need to be updated, then it only works on those 2 recipes with the X's. Otherwise, all the others are left alone. And if you're wondering kind of what a configuration file looks like, this is an example. So this is YAML format. Essentially, it's a very human-readable format and essentially specifies the input files. And we have essentially one configuration file per species. And so this is ultimately what's used to produce the output data file. And if you're maybe wondering at a high level, like what does this look like in practice when we're actually, when we've deployed the website? So essentially we have two containerized applications. So we have this pod on the left. So hopefully you can see my mouse. Yes. Which is the deployment, which is essentially the website, and that's continuously existing the entire time. So when someone interacts with the website, they interact with this this application. And then on the right, we have this job container, which every now and then is spun up and run and runs a job to essentially run make, so to rebuild the data sets if it needs to be updated. And the output from this make run is stored in what's called a persistent volume, which you can essentially think of as like an external hard drive. So the data is updated in this external hard drive every time we run this job. Then this external hard drive is shared across both containers. So this website can essentially directly access this data in the hard drive or in this persistent volume, which makes for a very good experience so far. And with that, I'd like to pass it on to Angela.
All right. Thank you very much for the wonderful introduction. I hope you are all motivated and identify some key features that could be useful in your work. Now I'm going to talk a little bit about things that we identified that could definitely be helpful for researchers to boost the way of work and facilitate, in general, how to share and communicate data. So essentially, why should we be so focused on visualizations? Well, we are, as primates, we are very interested and very visual, and indeed, genomic visualizations play a key role in life sciences, essentially for exploration purposes, to try to make sense about data, to interpret it in a essentially in a combined way, because this is actually the power of a genome browser, where, as is displayed here in the picture, you can essentially, in one screen, you have different data types, and then you have the capacity to compare across them and maybe find interesting patterns. For instance, like as Daniel was referring to, if we have a metric here that is telling us about genetic differentiation, and then you see that there is some complementary information, for example, in terms of gene expression that coincides with the position of these indication of genetic differentiation, then this could lead us actually to propose hypotheses and communicate discoveries. So that's why it's so important, and that's why we want to address this need in the community through the Genome Portal. And there are many, many different use cases for these kind of visualizations. One of them, for example, when people do comparisons of normal versus diseased individuals, then you can essentially combine different data types and then try to make inferences about potentially what could be the genotypic difference between different phenotypes, in this case, if it's a normal or a diseased individual, or populations distributed at the end range of an environmental gradient, and try to identify which could be genetic differences behind these phenotypic differences. So I'm just going to summarize some of the key benefits for researchers that we hope you take advantage of by accessing our service. One of them is, as Daniel mentioned, this is an aggregator. Essentially, we are an entry point for researchers in Sweden that essentially want to learn what is out there. So, of course, there might be instances where people go directly and find, look for the species of interest. But in other cases, people might be just browsing and, curious to learn about other projects ongoing in Sweden. So by bringing this graded exposure of your work and increasing this visibility, that could definitely be helpful for you as a researcher, and hopefully translate in, further collaborations or increase the number of citations of your work. Another beneficial part is by using the Genome Portal is to actually unlock genome data visualizations. For people that are bioinformaticians and very computer skilled people, I mean, you can easily install software and so on, but let's accept that not everyone has that skill set. So essentially, that's one of our core principles. We want to make genomic data visualizations accessible to everyone, regardless of the technical expertise. So indeed, we provide a URL, essentially a web address that you can share, and then you can actually add that URL to data availability statement of your manuscript. We require that all the data that is displayed in the Genome Portal is publicly available in a repository. However, we understand, of course, the use cases that, for example, might be a researcher working already in a manuscript and maybe would like to add this URL, the data availability statement. So in those cases, we can definitely accommodate that need by we only require that the data it's placed under embargo, essentially, in a public repository, and that the data has a accession number or a DOI, persistent identifier, and then we actually could start building their web page for you, even before the manuscript is already, or the data is already public. We can accommodate essentially both those cases, or also help you to make your data available. Another benefit is actually you will save yourself valuable time, because then you don't need to install software, you don't need to download large files on your computer over and over again, you or your team members, essentially. So all the data, as was explained before, we save a copy of it, we fetch that information, the data from public repositories, and basically the data is just at your fingertips, available on the internet. Of course, as many of you know, there are already many existing solutions on genome browsers, such UCSC, Ensemble, and so on. But so you might wonder what's the difference, basically with our service is that, often you need, if you want to address any requests in these large kind of like global initiatives, you need to wait. The staff maintaining those global services essentially might have different priorities, whatever. Here, the benefit is that you have a staff that's here in the room, essentially, that could help you to make your data available as soon as possible. So just expect that by going through this way, you might have a short waiting time to display your genomic data. Another nice feature about using actually JBrowse 2 is that you can export a publication quality image. You have the capacity to essentially go to a view, to a genomic region of interest, and then you can export that view as a SVG file. So this means that it's a vector file that you can easily edit in any graphic design software if you need, but that's one of the perks that come with this software. Another benefit is actually by using our service, we're going to improve the fairness of your data. As mentioned before, we want to promote sharing of genomic tracks or genomic annotations that rarely get public. They stay in researchers' computers, but we acknowledge that, often these data never gets public just because, not because it's not important, but it's mostly because it's not like mandatory for a publication process, but it's actually very, very valuable. In particular, those genomic annotations that, are derived from, comparative genomics analysis as conservation scores, or for example, population analysis, in which like you get genetic diversity metrics and so on. So by making your data more fair, also you're contributing to the wide research community and promoting data reusability, because ultimately that's the whole point about making our data findable, accessible, interoperable, and reusable. And it's like making the data not to us in the future, as researchers 6 months from now, we need to go back to the data, or maybe a year from now, but also hopefully this data that, it was generated with a lot of effort could be also used by others. And lastly, by using the Genome Portal, and particular JBrowse, you can enhance your teamwork. There are different features that are very, very nice. For example, you can share a session, you can share a view, you can actually bookmark the specific genomic regions that you might be interested to look after and you want to discuss with your colleagues. So are we doing with time?
Good.
Yes, okay. So as we're doing good in time, I'm just gonna quickly show you how it looks. But essentially, say we're in this session. So you can share the session just by clicking here on the share button, and you can easily copy to clipboard this URL, and then that's all. Then you share it with your teammates, and when that person wants to look at that, just like copy and paste that URL to the web browser. Feature that I'm very, very excited about is like you can bookmark regions. Say, I want to look at this region, bookmark, and then it's going to be displayed here in this bookmark region panel, and say, actually, I want to later look at this one. Bookmark, and then say, this one also looks super cool. Bookmark. And then you can actually annotate those bookmarks and say, oops, sorry. So I can now using my mouse. Yes, so say, I want to say, nice for later. And then you can add another label here for person 1 and whatever. Essentially, you can add little comments here and also okay, and then you can edit actually, I want this one to be highlighted in yellow. And this other one highlighted in green. So you can actually customize your views. And then you don't lose track of what you are looking at. And then say, I want to share this. You can either export it as a VED or TSV file. Then you can later share with your teammates. So it's a table essentially listing the regions of interest, or you can share it as a URL, and then essentially you first need to share the session, which is the genome, essentially the genomic data, and then share the bookmark, and then someone else can like upload both, and then they will by uploading those URLs, they can actually look at these exact same genomic data and the bookmarks that you tag. So for the sake of time, perhaps I will just go back and end by sharing that there are many more advanced features in JBrowse that we have time to discuss today, but essentially, I highly encourage you to take a look at the website. You can, visualize the structure variations, variants, high-C contact maps, quantitative tracks, and much more. Also, there are additional tools and functionalities through by the community or the developers of the genome browser. And I hope that you're interested in contributing and putting your data in our genome portal. So here's just a little reminder that it's as difficult as just sending us an email. And we start the conversation. Essentially, we're here to help. And there are actually 2 entry points to access the genome portal. One is directly using our URL, or another one is through the SciLifeLab Data Platform, which is the hub for data-driven life science services, data and support. So you can use either or both. We are listed there. You just need to go to services and find our name and then click, and then it will just connect it to our website. And with this, I want to acknowledge to a lot of people that we have time to or space to list here, but we are very thankful with everyone that helped us at any shape or form. In particular, I know this is not very long on my side to put people on the spot, but we are very thankful with all the researchers, collaborators that support this pilot implementation of the genome portal. You have them listed here. Your researchers were the shining light here, help us a lot to figure out different questions we had and also make this service possible. Thank you to staff at NBIS who provide us assistance data sets. Thank you to our funders, host institutions, and thank you for listening. So good. So now we open the floor for questions. Do we have questions in the chat?
Yes.
If you're not going to stop sharing,
Thank
maybe
you
we can
so
stop
much, Angela.
recording.
Yeah.